Have you ever wondered how a microbiologist identifies an unknown bacterial sample and finds the cause of an infection? Identifying bacteria is essential for prescribing the right antibiotics and stopping infections from spreading in hospitals and communities. This process isn't a single test, but a series of methods that together build a clear picture of the organism. Today we'll break down the typical workflow: isolation, microscopy and staining, biochemical testing, and molecular identification. Understanding these steps helps clinicians choose effective treatment and helps public health teams control outbreaks quickly. Let's start at the beginning: how samples are collected and how isolates are obtained in the lab. Proper sample collection is critical. Contamination or poor sampling can lead to misleading results and wrong treatment. Once received, samples are plated on appropriate media to isolate colonies. Each colony can represent a single species. Colony morphology, shape, color, edge, and texture gives initial clues and helps prioritize further testing. A pure colony is then subcultured so that subsequent tests analyze a single organism rather than a mixture. Once isolated, we move to microscopy and staining to learn about cell shape and structural features. These basic lab steps set the stage for targeted biochemical and molecular tests that confirm identity. Microscopy provides immediate information. Cell shape, foci, bacilli, spirilla, narrows the field of suspects. The Gram stain divides bacteria into gram-positive and gram-negative based on cell wall structure, guiding antibiotic choices. Gram-positive organisms retain crystal violet and appear purple. Gram-negative organisms appear pink after counterstain. Other stains, like acid-fast or capsule stains, highlight special features useful for more specific identification. Microscopy is fast and inexpensive, but it rarely identifies species alone. It directs the next set of tests. Next, biochemical tests probe metabolic traits that are often unique to certain genera or species. Biochemical tests assess how bacteria metabolize sugars, produce enzymes, or react with substrates. Results are often visual color changes. Common assays include catalase and oxidase tests, sugar fermentation panels, urease, and hydrogen sulfide production. Automated systems can run many biochemical tests at once and compare patterns to databases for likely species matches. Results generate a biochemical profile. Technicians compare that profile to reference tables to suggest probable identities. Phenotypic methods are powerful and cost-effective, but closely related species can share traits and require further testing. When ambiguity remains, molecular tests provide definitive identification by analyzing genetic material. Molecular methods like PCR and sequencing detect species-specific genetic markers and can identify organisms rapidly and accurately. DNA sequencing reads the organism's genetic code and matches it to reference databases to confirm species with high confidence. Molecular tests are especially useful for slow-growing or hard-to-culture organisms and for outbreak tracing. All test results, microscopy, biochemical profiles, and molecular findings are integrated to produce a final laboratory report. Clear reporting includes organism name, susceptibility recommendations, and comments about test limitations and next steps. In short, bacterial identification uses a stepwise approach combining culture, microscopy, biochemistry, and molecular tools to guide treatment and protect public health. Thank you for watching.